beyond the second messenger to phosphoprotein messengers. There are two major targets of signal transduction. These are phosphoproteins and genes. Many of the intermediate targets along the way to the genes are phosphoproteins, such as the fourth messenger phosphoproteins, that lie dormant in the neurons until signal transduction wakes them up and they spring into action. One signal transduction pathway can activate a third messenger kinase through second messenger cyclic AMP, whereas another signal transduction pathway can activate a third messenger phosphatase through second messenger calcium. In the case of kinase activation, two copies of the second messenger target each regulatory unit of the dormant or sleeping protein kinase. When two copies of cyclic AMP bind to each regulatory unit, the regulatory unit dissociates from the enzyme. The dimer dissociates into two copies of the enzyme and the protein kinase is now activated. The activation of a third messenger kinase adds phosphatase to a variety of phosphoproteins such as ligand-gated ion channels, voltage-gated ion channels, and various regulatory enzymes, which all can act as fourth messengers. Meanwhile, the nemesis of protein kinase is also forming, namely a protein phosphatase. Another first messenger is opening an ion channel here, allowing second messenger calcium to enter, which activates the phosphatase enzyme calcineurin. In the presence of calcium, the calcineurin becomes activated, and it's ready to rip phosphatase group of the fourth messenger phosphoproteins, such as the ligand-gated ion channels, voltage-gated ion channels, and the various regulatory enzymes. The activation of this fourth messenger can change the synthesis of neurotransmitters, can alter neurotransmitter release, can change the conductance of ions, and generally maintain the chemical neurotransmission apparatus in either a state of readiness or dormancy.